Please go ahead. Here is
Okay.
Julian and Michelle presenting their summer internship project.
So I'm Julianne Go.
And I'm Michelle.
And today we will be discussing our findings over the summer on the role of the Dynorphin Kappa opioid receptor system in alcoholism.
So first we'll begin with statistics. So alcohol use disorder, also known as AUD, which can be alcohol abuse, dependence, addiction, and tolerance, is a major problem here in the United States. Between 2006 and 2014, the rates of all alcohol-related emergency department visits grew by 47%, resulting in a yearly average increase of 210,000 alcohol-related ED visits. The 2019 National Survey on Drug Use and Health, or the NSDUH, found that 14 million people ages 12 and up have AUD. Nearly 40% of high school seniors have either smoked cigarettes and or marijuana, 25% have used illicit drugs, and 70% of them have consumed alcohol. And more recent data shows that 39.7% of 12 to 20-year-olds admitted to having at least one drink in their lifetimes, showing how as time progresses, the age at which the young consume alcohol decreases. This is due to the fact that adolescents appear to be less sensitive to the aversive properties of drugs of abuse as compared to adults. And data from 2016 shows that alcohol abuse was the 7th leading cause of premature death and disability worldwide. And in the United States alone, it is now the 3rd leading preventable cause of death. And then on to effects on the body. The effects of alcohol differ from person to person depending on a range of factors, such as how much they drink, how frequently they drink, their age, their health state, and their family history. People who drink excessively over a long period of time may suffer from the long-term effects of alcohol, which can include AUD, health difficulties, and increased risks of certain cancers. One of the most common effects is withdrawal and the symptoms that accompany it. Withdrawal is one of the distinguishing symptoms of alcoholism and is frequently characterized by a decrease in the physiological function and increased negative effects. Increased anxiety and depression have also been seen to occur. Additionally, the Winnick-Korsakoff syndrome can also occur in a person who has long-term alcohol abuse. And moreover, many parts of the body can fall victim to the harsh effects of alcoholism, such as the heart, the liver, and the dopamine system. Now on to Julian with age impacts.
So adolescence is a stressful yet key period for brain development in humans and is also a period in time when individuals are the most reckless and impulsive. Thus, adolescents are more susceptible to alcohol use and abuse. When adolescents consume alcohol at a young age, they don't experience the aversive effects of alcohol that adults do. As they grow, these people require more and larger doses of alcohol in order to experience the same reward as when they were younger. Therefore, alcohol consumption at a young age can lead to alcohol use disorders in adulthood and chronic alcohol intake. Insensitivity to many adverse ethanol effects, such as sedative and anxiolytic relative to adults, may serve as a fact for increased ethanol consumption during adolescence. Alcohol abuse and alcohol disorders also have a significant socioeconomic impact as a rising public health issue. Overconsumption of alcohol due to its addictive nature can lead to compulsive alcohol taking and seeking behaviors. Despite its prevalence and commonality, there are only a few pharmacotherapeutic agents available to treat alcohol disorders, partly because there is a lack of overall understanding of the specific neurobiological mechanisms that outline the rewarding actions of alcohol consumption. The annual social expenses of alcohol use disorders are estimated to be at least $148 billion. Clearly, there is a need for more research and treatment of alcoholism, and advancement in this field would be highly helpful to society as a whole to decrease the cost to society and help those suffering from alcohol use disorders. So the other opioid peptides and the respective receptors besides the dynorphin KOR that play a role in alcoholism and the reward system of alcohol consumption are enkephalines that correspond with the delta receptor and endorphins that correspond with the mu receptor. Endogenous opioids are linked to a variety of functions, including ethanol reward and reinforcement, and several lines of evidence suggest that increased opioid activity in response to acute ethanol exposure mediates ethanol intake and preference. The enkephalin delta opioid receptor system is involved in the rewarding, reinforcing properties of drugs of abuse, as it modulates presynaptic release of dopamine in the ventral tegmental area, as well as the nucleus accumbens. This is significant because these two brain regions are associated with the development of addiction. Although there has been conflicting results about the reinforcing effects of the enkephalin delta opioid receptor system in addiction, antagonists such as naltrindol and naltrexone have shown to successfully decrease alcohol consumption in mice. The enkephalin delta opioid receptor system has also been linked to effects that follow chronic use of opiates, which include stress, inflammation, and anxiety. Additionally, the endorphin mu opioid receptor system is credited with mediating the acute analgesic effects of opiates. Similar to the antagonists of enkephalin and dynorphin, antagonists of endorphins have shown to decrease ethanol CPP in mice compared to those not administered the antagonist, which is naltrexone.
So here's kind of a graph that shows all the different relationships that Julian just mentioned. And in our specific presentation and research that we've done over the summer, we specifically focused on dynorphin and KORs, or kappa opioid receptors. So dynorphin is a complex neuropeptide whose precursor is prodynorphin. Prodynorphin binds really well with kappa opioid receptors and is known to downregulate dopaminergic tone, and also produces aversive states that may affect the development of opioid dependence. Dynorphin production is located throughout the brain, with kappa opioid receptors located in the cortex, amygdala, stratum, thalamus, hypothalamus, and the VTA, or the ventral tegmental area. This opioid peptide is made up of amino acids and exists in many forms, including dynorphin A and dynorphin B.
So moving on to an overview of the DYN-KOR system. Although less studies, other data suggest that this system may play a role in either ethanol's unpleasant characteristics or ethanol-induced negative reinforcement. Increased activation of the DYN-KOR system would be predicted to limit ethanol consumption, whereas pharmacological inhibition of KOR would be expected to increase ethanol consumption. Several lines of evidence imply that the positively reinforcing and stimulatory effects of ethanol are connected with activation of MOR and potentially DOR, whereas the unpleasant effects of ethanol administration are associated with the DYN-KOR system. Additionally, studies have investigated the effects of kappa antagonists on ethanol consumption and self-administration, but resulted in mixed results, with both enhancing and suppressing effects of it being reported. Studies have also shown that the DYN-KOR system's importance in ethanol consumption in relation to dependence and stress-induced elevation in intake. Mu opioid receptors are seen to have opposing effects as KOR, as these receptors cause highly reinforcing results and increased dopamine release, while activation of the KOR results in aversive or dysphoria states and decreases in dopamine release. Locomotor behavior, feeding behavior, and sexual behavior are all also impacted by the activation of KOR.
Now on to different alcohol self-administration and consumption methods. So for many studies involving self-administration, mice are usually given a two-choice bottle paradigm. One study proposed that if there was an increase in KOR signaling, or the DYN system is activated following chronic alcohol exposure that is significant enough to result in increased self-administration of alcohol, KOR agonists should be successful in reducing the negative states associated with withdrawal to reduce the excessive alcohol self-administration. Another study that used the two-choice paradigm showed how the deletion of the pre-prodynorphin gene in female mice causes a significant decrease in alcohol consumption preference. Additionally, prodynorphin activation may be required in mice, which makes a connection between palatability and consumption. There was also a similar study conducted to the one just mentioned above, this one now using KOR knockout mice, in order to examine the effects of opioid receptors on oral alcohol self-administration. The disruption of the DYN-KOR affects sensory rewards through the central mechanism, which reduces alcohol palatability. The ability of the KOR blockade to regulate the ethanol-evoked dopamine release in the nucleus accumbens leads to the conclusion that a decrease in the activity of KOR systems may lead to an enhancement of the reinforcing effects of ethanol. So another form is the sweetener fade method. Alcohol self-administration has been used to test if the intracentral nucleus of the amygdala antagonist-mediated reductions could be dissociable from changes in physiological withdrawal. For this study, alcohol self-administration was conducted using the sweetener fade method, which meant that over time, the solution of sweetener and alcohol would be replaced with an alcohol-only solution. Escalated self-administration of alcohol persisted into protracted abstinence and was dose-dependently rescued by the intra-central amygdala norepinephrine fusions. The study indicates that in the central amygdala, KOR antagonists' efficacy for reducing escalated alcohol self-administration is dissociable from KOR antagonist effects on the physiological withdrawals. The study also suggests that the dysregulation of the central amygdala DY-KOR system endures and continues to drive escalated alcohol self-administration.
So KOR agonists and antagonists were often used in studies to determine the effect of on alcohol consumption. Antagonists are seen to decrease alcohol self-administration, whereas antagonists are seen to activate the DY-KOR system. Additionally, norepinephrine, a KOR antagonist, has been seen to decrease ethanol self-administration in alcohol-dependent subjects, but had no effect on non-dependent subjects. One short-acting KOR agonist, cyclophane, was used to test the efficacy in decreasing alcohol self-administration in alcohol-dependent rats and mice. U5488H, a long-acting KOR agonist, was also used in the study. Cyclophane was seen to significantly block KOR-induced effects, but had no effect on self-administration of alcohol for vapor and air-exposed animals. Overall, in the mouse brain tissue, selective KOR antagonist effects were found caused by cyclophane through reduction of U5488H stimulated coupling at lower concentrations. However, in the rat brain, cyclophane did not affect U5488H stimulated signaling at any concentration. Moving on to opioid regulation of the anxious and depressive states, various models are available to investigate anxiety and depressive-like behavior of the DY-KOR system in relation to alcohol. Many studies have been conducted in order to observe how alcohol consumption induces or relates to feelings of anxiety or depression. Some models used in these studies include the elevated plus maze, the light dark box, the forced swim stress test, and assessing shelter-seeking and risk-taking behavior, which are all useful in assessing anxious and depressive behavior. Important findings found by studies who use models mentioned before include prenatal ethanol exposure alters gene expression of the prodynorphin KOR system, which is correlated with an anxiety-prone phenotype at the behavioral level. Norepinephrine.
Okay, go ahead.
So norepinephrine reversed behavioral changes in rats during an acute ethanol withdrawal and protracted ethanol abstinence. Depressive-like behavior may be enhanced during abstinence, even if motivation and anxiety-like behaviors disappear. The long-term alterations to the DY-KOR system are likely involved in regulating the depressive-like states associated with ethanol dependence. Interactions between the DY-KOR system and other stress-related neurotransmitter systems may explain why norepinephrine reduces the effects of a mild external stressor in rats with a history of ethanol dependence, while low doses of U5488H do not induce anxiety-like behaviors in ethanol-exposed rats. The DY-KOR system is a critical mediator in the stress-related effects of alcohol, and while KOR antagonism may be sufficient to reduce the heightened stress response found after long-term ethanol withdrawal, direct activation of the DY-KOR system may not be required to elicit stress-related behaviors. The DY-KOR system is involved in regulating an anxious response during ethanol withdrawal, as norepinephrine significantly decreased open arm exploration in one study. Anxiety-like behavior associated with ethanol withdrawal is blocked by KOR antagonism, and results suggest that similar mechanisms are involved in regulating the changes in behavior that are associated with ethanol withdrawal and induced by KOR agonists. Ethanol-induced CTA following a stress exposure depends on age, and KOR antagonists only reduce aversive effects of ethanol in stressed adults. When the KOR system is already activated due to stress, there's involvement of the system in adult animals in the ethanol aversive properties. The typical enhanced sensitivity to drug reward found in adolescents may be due to the increased stress sensitivity during this developmental period, which is a time in which individuals experience stress that may activate the DY-KOR system.
Now on to studies of increased alcohol consumption in different brain regions. So different brain regions have been implicated in the activation of the DY-KOR system with relation to alcohol. When investigating the effects of alcohol consumption on different brain regions, there are several techniques that can be deducted in order to see these effects. One such study used pharmacological and chemogenic techniques to see that binge-like alcohol consumption is mediated, at least in part, by DYN-KOR signaling in the central amygdala. One experiment conducted in this paper looked into the effects of the intracentral amygdala injection of KOR antagonists on binge-like alcohol drinking. During the drinking in the dark or DID treatment, mice consumed alcohol every week. The mice were then given microinjections of norbNI into the central amygdala, which was shown to reduce alcohol consumption. These findings demonstrated that the direct pharmacological inhibition of KOR in the central amygdala reduces alcohol consumption, implying that KOR signaling within the central amygdala plays a role in regulating alcohol consumption. Moreover, this study is important as its findings give additional evidence for the function of the DYN-KOR system activity, specifically activation of this neuropeptide system in the central amygdala. And then another study was used to use systematic injections of ethanol to observe that it allowed for the release of beta endorphins in the midbrain VTA in a dose-dependent way, with low to medium but not high doses of ethanol enhancing beta endorphin release. This ethanol-induced increase in beta endorphin release in the ventral tegmental area of the midbrain could play a key role in the ethanol-induced stimulation of the mesolimbic dopaminergic system and its commencement of alcohol reward and its reinforcing processes.
Moving along, there are differences in the activity of numerous components of the endogenous opioid system under basal conditions and after ethanol exposure between strains and lines of animals that consume either a lot or little ethanol. And because of that, one paper sought to determine whether there were variations in the density of kappa opioid binding sites, the content of pro-dynorphin mRNA, and the amount of dynorphin peptides in different brain areas of mice. The two types of mice that were used were the C57BU6 mice, which were ethanol preferring, and DBN2 mice, which were ethanol avoiding. Because of increased stimulation of kappa opioid receptors at the nucleus accumbens has been linked to decreased dopamine release in aversive states, the higher content of kappa opioid receptors, pro-dynorphin mRNA, and dynorphin peptides in limbic system regions of DBN2 mice may play an important role in determining why they consume less alcohol. Similarly to the paper referenced just before this, another paper explored how different brain regions were affected in terms of neuropeptide levels. In order to do so, the opioid and nociceptin orphanine FQ systems, as well as the opioid peptides dynorphin B and MEP, were studied in alcohol-preferring C57BL6J mice. Results show that because there are strain differences between C57BL6J mice and DBA2J mice, it may lead to diverse drug-taking behavior, as well as time and structure-specific changes in neuropeptide levels after alcohol self-administration in C57BL6J mice.
Additionally, microdialysis studies are quite universal and can be used in a myriad of experiments to look at different aspects of how alcoholism can change peptide levels. The goal of one such study was to analyze the effects of alcohol administration, small, medium, and large doses, or a stress-induced event on the levels of dynorphin A 1 through 8 in the nucleus accumbens of rats. The nucleus accumbens plays a key role in processes that involve stress and reinforcement. It was determined by this study that the increase in dynorphin levels in the nucleus accumbens depends on the alcohol dose. Rats with a low alcohol dose did not show any effects on the dynorphin A 1 through 8 levels, while rats given higher doses showed a significant increase in dynorphin levels. So now moving along to changes in peptide levels in mRNA, tissue content measurements, and microdialysis, the release of dynorphin in the nucleus accumbens because of an alcohol-induced stimulation may regulate the activity of other local transmitter systems to produce a stress state in an animal. Additionally, dopamine and glutamate releases in the nucleus accumbens stimulated by a low dose of alcohol usually does not exist with larger doses of alcohol. Changes in mRNA levels for dynorphin and KORs following the administration of alcohol or withdrawal of it have been observed. One study used brain tissue samples from those of European descent who were deemed alcoholics. Linear regression models were constructed using statistical analysis from the data found in the study. And the correlation between prodynorphin and OPRK1 expressions significantly differed between alcoholics for intoxication status and their controls. And then the slope of the regression line was steeper in alcoholics as compared to controls. This suggests that alcoholics with a high OPRK1 expression have greater prodynorphin mRNA levels than their respective controls. Because of increased dynorphin levels compared to the controls, alcoholics with high prodynorphin and OPRK1 expressions will exhibit a larger and stronger KOR activation. And then another paper dove into investigating the fact that there is an increase in KOR mRNA levels in the nucleus accumbens and BTA after the last dose of ethanol had been seen in rats who were repeatedly treated with ethanol. The downregulation of KORs and mRNA in these brain regions may suggest an adaptive response to the system to an increased activity in dynorphin nerve terminals. The activation of dopamine nerve terminals induced by ethanol may be counteracted by the DYN-KOR system. As such, repeated ethanol administration induced both immediate and long-term alterations in the tissue concentration of brain opioids. The dynorphin B levels were significantly elevated in the nucleus accumbens 30 minutes and 21 days after the last dose, as seen from analysis conducted from the results from experiments held in this paper, which suggests that repeated exposure to ethanol may lead to long-lasting alterations in the turnover of dynorphin in this brain region.
So alcohol has been seen to have different effects on behavior based on different factors, which is due to the DYN-KOR system. Differences in condition place preference, condition taste aversion, condition place preference, and condition taste preference, as well as between ages and sexes, have been observed by a variety of different studies. Important findings found by studies who observed these behavioral changes include mice who lacked the DYN gene displayed a greater CPP in the presence of ethanol compared to control mice. Sex and age differences in the propensity to develop ethanol CTA may contribute to age and gender inequalities in ethanol consumption. For example, adolescent insensitivity to the unpleasant effects of ethanol relative to their adult counterparts may allow for the higher ethanol intake levels reported during adolescence, particularly among males. Similarly, adult females may consume more ethanol than their male counterparts, possibly due to being less sensitive to the adverse effects of ethanol than adult males. Sensitivity to KOR agonists may depend on age, as there is a general pattern of reduced sensitivity in adults to agonists with both CPA and CTA paradigms. Reduced sensitivity to KOR agonists in adult and adolescent animals may be due to the DYN-KOR system not being fully developed, thus less likely to become activated. The effects of the KOR agonists were dependent on age and stress, with sex having a significant effect on one response measure. Activation of KOR suppressed social behavior at all ages in non-stressed rats, although sensitivity to the suppression varied with age. The DYN-KOR system might be more sensitive in females than males because of an increased susceptibility to psychopathologies that may contribute to the increased negative consequences to the abuse of drugs. Differences are apparent between juveniles compared to adolescents and adult rats in responsiveness to a stress event. In terms of stress-induced behavioral alterations and stress-associated changes in the social consequences to the DYN-KOR system activation. Decreases in responsiveness to pharmacological KOR activation in adults compared to juveniles suggest an ontogenetic increase in stress-induced activation of the DYN-KOR system. The ability for the KOR blockade to impact ethanol consumption varied based on age, as adolescents of either sex were seen to not have a significant difference in ethanol intake regardless of dose or of NOR-BNI. Males and females have a different response to KOR manipulation and drug-related behaviors. Adult males have a significant control over ethanol intake because they are extremely sensitive to the aversive properties of KOR when the DYN-KOR system is activated by ethanol. Adolescents not being affected by the antagonism of the KOR system as adults may again be due to the DYN-KOR system playing less of a role in ethanol intake regulation during adolescence due to a decreased sensitivity to the KOR system.
So for one of our projects, we created a CPP model of the experiments that would be conducted. So there's usually a preconditioning day where rats are able to explore all the different regions. One side is usually a saline paired chamber, the other side being an ethanol paired chamber, and the central chamber is the neutral one. And then following preconditioning days are conditioning days, in which usually in a morning session, rats are placed into one of the chambers, and then in the afternoon, they switch the chambers. And then following that are post-conditioning days, where mice are given free rein to explore and then to see if they prefer any chamber. And then in this one specific paper, they had an additional post-conditioning day in which they recorded any long-lasting effects.
So in conclusion, the DUIN-KR system plays a very important role in regulating the negative symptoms of increased alcohol tolerance, abuse, and addiction. The system has been found to be involved in regulating the depressive-like states associated with alcohol dependence. Moreover, KORs have been seen to regulate stress and other anxiety-inducing events caused by alcohol withdrawal. Data shows that different kappa agonists and antagonists can help increase alcoholism in both long-term and short-term addiction. Moreover, this system is key in alleviating the negative affective states seen in both adolescent and adult alcoholics. Studies above show how targeting the DUIN-KR system directly impacts alcohol withdrawal symptoms. As such, with all of the information obtained through experimental procedures done on different species of both rats and mice in many literatures, it can be used to help human alcoholics on their way towards recovery as a form of treatment. Data backs the claim that kappa agonists and antagonists such as NORBI9 and U69 are successful in reducing the intake of alcohol on animals and thus in humans as well. This form of pharmacotherapeutics can be most essential in the fight against increasing alcoholism here in the United States and elsewhere.
Thank you for listening to our presentation.
Thank you. Excellent job. Let me just stop recording.